Hey guys, welcome back. Today's topic is modes of reproduction used by single organisms. This is from class 10 biology. Let's begin the session. Here, reproduction is a biological process by which living organisms produce young ones of their own kind. And the importance of reproduction is it enables the continuity of species on Earth. Now, in today's session, I'll be discussing about the modes of reproduction used by single organisms. The modes by which various organisms reproduce depend on the body design of the organism. This is one very important statement. I repeat this once again. The modes by which various organisms reproduce depend on the body design of the organisms. Here, asexual reproduction is common among single-celled organisms and in plants and animals with relatively simple organizations. So, here the single-celled organisms have simple organization, and also some of the plants and animals have relatively simple organizations. In such organisms, asexual reproduction is found. Now, moving to the continuation here, what is asexual reproduction? Here, in this method, a single individual, that is parent organism, is capable of producing offspring. So these offspring are the young ones. Okay, so in asexual reproduction, single parent organism, single individual is capable of producing offspring. These offspring are nothing but the young ones. Now, as a result, the offspring that are produced are not only identical to one another but are also exact copies of their parent. So, the offspring which are produced by this asexual method, they are not only identical to one another but are also the exact copies of their parent. So, here one important term is used, that is what we call clone. Okay, so the term clone is used to describe such morphologically and genetically similar individuals. So, how can we define the word clones? The answer is the morphologically and genetically similar individuals are called clones, and clones are produced by asexual reproduction by single parent organism. Here, morphological means the external features, okay, and genetically means the genetic material, the DNA. It is related to the DNA. So, they are similar in such individuals. So, morphologically and genetically similar individuals are called clones, and they are produced here during the asexual reproduction. Now, moving to the continuation, here there are different methods of asexual reproduction, and here now we are going to discuss about one by one these different methods of asexual reproduction. Here, one of the method is fission. Here, for unicellular organisms, cell division or fission leads to the creation of new individuals. Many bacteria and protozoa simply split into two equal halves during cell division. So, this is a very simple method of reproduction. Okay, so here many of the bacteria, the protozoans, simply split into two equal halves during cell division, and this results in the formation of new daughter cells. So, this is a mode of asexual reproduction. Here, one single parent organism is involved. and many different patterns of fission have been observed. Okay, and now we are going to discuss about what are the different patterns of fission seen in organisms. Here, first of all, let's discuss about binary fission. This is one of the pattern, and here in organisms such as amoeba and some other organisms like paramecium, the splitting of the two cells during cell division can take place here in any plane. So in case of amoeba, it is any plane, but in case of other organisms which have a definite shape, it is a particular direction. In a particular direction, the plane of division will divide the cell into two daughter cells. But in case of amoeba, as amoeba do not have definite shape, okay, there is no definite shape in amoeba. So amoeba divides into two daughter cells in any plane. It can be in any plane. It can be like this, or it can be like this, or like this, or like this. In any direction, the cell can be divided into two equal halves. Okay, so that is what we call irregular binary fission. Irregular binary fission, or it is also called simple binary fission. Irregular binary fission or simple binary fission is seen in amoeba. You can see here, this is the parent cell. Okay, so this is the parent cell of amoeba, and these are the daughter cells. And here what we need to remember is two important steps. One is karyokinesis. Karyokinesis is nothing but the division of nucleus. Okay, so the division of nucleus is called karyokinesis. Okay, and here the nucleus will divide into two daughter nuclei. You can see here, this is one single nucleus, and here you can see this is one and this is the another. So two daughter nuclei are formed. This is what we call karyokinesis. And after this, cytokinesis occur. Cytokinesis is nothing but the division of cytoplasm. After the karyokinesis, after the division of nucleus, the cytoplasm is divided into two, like this, and then it is distributed into the two daughter cells. So binary fission involves two important steps. One, karyokinesis. It is nothing but the division of nucleus. And the second, cytokinesis. This is nothing but the division of cytoplasm. Okay, so this is how the binary fission occurs in amoeba. And in amoeba we find irregular or simple binary fission. And in this mode of binary fission, the plane of division can pass through any direction, like it can be like this, or like this, or like this, like this. In any direction, the plane of division can divide amoeba into two daughter cells, and this is how two daughter cells are formed. This is about binary fission in amoeba. Now let's discuss about one more example. Here some unicellular organisms show somewhat more organization of their bodies, such as seen in case of Leishmania. This Leishmania, it causes Kala-azar. Okay, this is a disease, and Leishmania is a protozoa. It causes Kala-azar, and it has a definite shape. You can see here in this picture, it has a definite shape, and it has a whip-like, okay, flagella is seen. Okay, so you can see here the whip-like structure. This long flagella is seen here. So on one particular site of this Leishmania. So it has a definite shape, and it has somewhat more organization when compared with amoeba. Now here you can see how the cell is dividing. You can see here, this is the initial normal Leishmania cell. Now you can see how it is undergoing the binary fission. The next and the next. So as it is undergoing these changes, you can see here, this is one single nucleus you will find here. This is a single nucleus. But here it is dividing into two. So here also we find the same steps. Karyokinesis is the first step. Here the nucleus is dividing into two daughter nuclei. So this is how the nuclei are divided. Now after that, in the next step, obviously there will be cytokinesis. Cytokinesis is nothing but the division of cytoplasm, and this is how it is dividing in only one particular direction, in one single plane of division. The Leishmania is dividing into two daughter cells. You can see this is the plane of division. Okay, so here we find longitudinal binary fission. Okay, how the cell is divided? Here the Leishmania cell is divided like this in longitudinal direction. Okay, so this is the plane of division, and it is dividing the parent cell into two daughter Leishmania cells. So here in the somewhat more organization is seen in Leishmania, and so here a plane of division is passing through one particular direction, and that is longitudinally it is passing, and longitudinal binary fission is seen in Leishmania. So this is how the cell is divided into two, and two daughter cells are formed. So here the binary fission occurs in a definite orientation in relation to the structure. Okay, so this is about binary fission. Now moving to the next, here multiple fission. So this is, in this case, many cells are formed from one single parent cell, and here we see the example multiple fission in Plasmodium. Okay, so here the malarial parasite, the Plasmodium, divide into many daughter cells simultaneously by multiple fission. So here one single cell, the Plasmodium, okay, parasite, the malarial parasite, it undergoes multiple fission. And dear students, here the steps are quite different when compared with binary fission. Here karyokinesis occurs several times. Okay, this is one very important statement. So karyokinesis occurs several times, and here one very important aspect is, after every karyokinesis, cytokinesis do not occur. Instead, a bit of cytoplasm accumulates around each nucleus. Okay, so this is how you can see many cells are formed inside, but here after every karyokinesis, cytokinesis do not occur. Very important point that you need to remember. Instead, what is happening? Around each nucleus, a bit of cytoplasm accumulates, and then a membrane is formed, and this is how many daughter cells are formed inside. Okay, so this is about multiple fission in Plasmodium. Okay, so here many daughter cells are formed. Okay, that is the important aspect. And one more thing is, after every karyokinesis, cytokinesis do not occur. Instead, a bit of cytoplasm accumulates around each nucleus. Now moving to the continuation, one more example we speak here, multiple fission in amoeba. Under unfavorable conditions, this is important. Okay, during normal favorable conditions, amoeba reproduce by binary fission, but under unfavorable conditions, the amoeba withdraws pseudopodia. These pseudopodia are false feet seen in amoeba. So it withdraws the pseudopodia and secretes a three-layered hard covering called cyst around itself. So amoeba forms a cyst around itself during unfavorable conditions, and when favorable conditions return, the encysted amoeba divides by multiple fission and produce many minute amoebae or pseudo podiospores. Okay, so this is what we need to remember. So here I repeat this once again. Under unfavorable conditions, the amoeba withdraws pseudopodia and secretes a 3-layered hard covering or cyst around itself. Okay, and this step we can call encystation. This is the step. Now after that, when favorable conditions return, then the encysted amoeba divides by multiple fission and produces many minute amoebae or pseudopodiospores. So this step is what we call sporulation. Okay, so this is what we call sporulation. Okay, the cyst will burst out and the spores are liberated in the surrounding medium to grow up into many amoebae. So this is what happens in the later stages. So what happens here? The cyst bursts out and the spores are liberated in the surrounding medium. They grow up into many amoebae. So this is how multiple fission occurs in amoeba during unfavorable conditions. So till now we discussed about fission. We discussed about binary fission and multiple fission. And if we speak about the differences between these two, in binary fission, 2 daughter cells are formed. In multiple fission, many daughter cells are formed. In binary fission, every part of the parent cell is utilized. But in case of multiple fission, a bit of cytoplasm is not utilized, membrane is not utilized. So every part of parent cell are not utilized for the formation of daughter cells. Okay, and binary fission occurs during favorable conditions. Multiple fission occurs during both favorable conditions as well as unfavorable conditions. Okay, and in case of binary fission, every karyokinesis is followed by cytokinesis. But in case of multiple fission, every karyokinesis is not followed by cytokinesis. So this is about the difference between binary fission and multiple fission. Now let's proceed with one more concept, that is regeneration. Here many fully differentiated organisms. What is differentiation? You have learnt in lower classes. The cells attain definite shape, size and perform specific functions. They perform specific functions. Okay, that process is called differentiation. Okay, cells attain definite shape, size and they perform specific functions, that is differentiation. So many fully differentiated organisms have the ability to give rise to new organisms from their body parts. I repeat this statement once again. Many fully differentiated organisms have the ability to give rise to new organisms from their body parts. And here, for example, we speak about the simple animals like Hydra and Planaria can be cut into any number of pieces and each piece grows into complete organism, and this is what we call regeneration. Very, very important statement. I repeat this once again. Here, simple animals like Hydra and Planaria can be cut into any number of pieces and each piece grows into a complete organism, and this is regeneration. That is, if the individual is somehow cut or broken up into many pieces, okay, then many of these pieces grow into separate individuals. You can see here this picture. This is regeneration in Planaria. Regeneration in Planaria. This is a parent organism. Okay, so this is parent organism. You can see here somehow it is broken down into 3 pieces. Okay, now you can see that each piece is developing the remaining part of the body. You can see this is head region, it is developing the remaining part. This is the middle portion. It is developing both the upper and lower parts, and this tail region is developing the upper part. As a result, here you can see the complete planaria organisms are formed. So this is how, if an organism is somehow broken down into many pieces, then each piece is giving rise to a new organism. This is what we call regeneration. The new organisms are formed from parts of the body. So this is one of the very important question for board exams, and this diagram is also very important for 3 marks. Okay, now moving to the continuation here, what actually happens, what is the mechanism involved in regeneration process? Let's discuss. Here regeneration is carried out by specialized cells. Okay, so these specialized cells are totipotent cells. Okay, I repeat this once again, very important concept. This regeneration is carried out by specialized cells, and these cells proliferate and make large number of cells. Okay, these cells have the ability to produce large number of cells, all types of cells. Okay, so from the mass of cells, different cells undergo changes to become various cell types and tissues. So this is what we call differentiation. Okay, so here from the mass of cells which are formed from these specialized cells, what happens? Different cell types, they undergo changes, they become various cell types and tissues, and these changes take place in an organized sequence which is referred to as development. Okay, so all these events which are occurring during the regeneration process, they take place in an organized sequence which we refer as development. Okay, and however, we should not get confused. Regeneration is not same as reproduction, since most organisms would not normally depend on being cut up to be able to reproduce. So we should not say that regeneration is same as reproduction. Actually, regeneration is not same as reproduction. It is not same as reproduction, because most organisms would not normally depend on being cut up to be able to reproduce. So that is what we say about regeneration, and this is how the organisms like planaria and the organism like Hydra, they undergo regeneration process to produce the young ones in large numbers. So this is what. Now moving to the next, one more important concept is fragmentation. So here in multicellular organisms with relatively simple body organization, so here relatively simple body organization is seen, and simple reproductive methods can still work. I repeat this statement once again. So in multicellular organisms with relatively simple body organization, simple reproductive methods can still work. Example, Spirogyra. Okay, so this is an alga. Okay, this is an example of alga. Okay, it simply breaks up into smaller pieces upon maturation, and these pieces or fragments grow into new individuals. I repeat this statement once again, very important. So here Spirogyra, for example, simply breaks up into smaller pieces upon maturation, and these pieces or fragments grow into new individuals. You can see here, this is the picture of a Spirogyra, okay, and alga. Okay, here this is a filamentous structure. Now if it is divided into pieces like this, here suppose if we divide it, if it is divided here, then you can see if it is fragmented like this, if it is cut like this into fragments like this, okay, then each fragment will form a new Spirogyra. So from these pieces or the fragments, new Spirogyra are formed if it is splitted into different pieces. Okay, so this is what we call fragmentation in Spirogyra. So it results in formation of new Spirogyra. So this is actually relatively simple body organization. Spirogyra has relatively simple body organization. It is actually thallus. It is undifferentiated body. Okay, so because it has a relatively simple body organization, simple reproductive methods like fragmentation can still work in Spirogyra. Okay, now moving to the continuation here, all multicellular organisms cannot simply divide cell by cell, as many of them are not simply a random collection of cells. Important statement. I repeat this once again. All multicellular organisms cannot simply divide cell by cell, as many of them are not simply a random collection of cells. Here, specialized cells are organized as tissues. It means cells are organized to form tissues, and tissues are organized to form organs, which then have to be placed in definite position in the body. This is what a complex organization is seen in many multicellular organisms. Okay, so here in such a carefully organized situation, cell by cell division would be impractical. Okay, and multicellular organisms therefore need to use more complex ways of reproduction. Okay, I repeat this statement once again. In many multicellular organisms, what do we find? Complex organization. Specialized cells are organized to form tissues, which are organized to form organs, and they are placed at definite positions in the body. So because of this complex organizations that we find in multicellular organisms, okay, so cell by cell division would be impractical, and multicellular organisms therefore need to use more complex ways of reproduction. Okay, so complex multicellular organisms use more complex ways of reproduction. Now here a continuation part. A basic strategy used in multicellular organisms is that different cell types perform specialized functions. This is what we call differentiation process. Following this general pattern, reproduction in such organisms is also the function of specific cell types. So reproduction is also the function of specific cell types in complex multicellular organisms. Okay, now moving to the next, here one more method of reproduction is budding in yeast. Okay, here in yeast, small buds are produced that remain attached initially to the parent cell, which eventually gets separated and matured into new yeast organisms. These are the cells actually. Yeast is the unicellular fungi. Okay, it is a unicellular fungus, and here budding is a mode of asexual reproduction. You can see here this picture, you will understand better. This is a parent cell. This is the parent cell, yeast. Okay, you can see here small bud is arising from the parent cell. This is how it will grow, it will develop, and then it detaches from the parent cell. Like this it will detach, and then it will grow as a separate organisms. Okay, and in yeast, here also a chain of buds can be formed like this. Okay, so this is also seen in case of yeast. This is what we call chain of buds. So here in case of yeast, small buds are produced that remain attached initially to the parent cell. You can see here these are remaining attached initially to the parent cell. And then what happens? Eventually they get separated and mature into new yeast organisms. They are the new cells because yeast is a unicellular fungus. Now, budding is also seen in other organisms like Hydra. This is Hydra. Okay, so budding in Hydra. Here in Hydra, a bud develops as an outgrowth due to repeated cell division at one specific site. You can see here, this is one specific site. This is one specific site, let's assume. Okay, at this specific site, because of repeated cell division, here a bud develops. And this bud, okay, develop into tiny individuals. When they are fully mature, detach from parent body and become new independent individuals. You can see here, because of repeated cell division at one particular site, here at this specific site, a small bud is arising. And this small bud, it grows, it develops. When it is matured like this, it detaches from the parent body. It leads a separate life. Okay, it leads a separate life after detachment from the parent body. So this is how budding in Hydra occurs. And here organisms like Hydra, they have specialized regenerative cells, specialized regenerative cells. So because of these specialized regenerative cells, at specific sites, buds are formed because of repeated cell division. And this bud is gradually growing. Once it is matured, it detaches from the parent body and it leads a separate life. So this is how budding in Hydra occurs. So today we have discussed different modes of asexual reproduction. We have discussed about the modes of reproduction used by single organisms. Okay, so here initially we discussed about asexual reproduction by fission and the various methods like binary fission in amoeba, which is irregular or simple binary fission. Then we discussed about longitudinal binary fission in Leishmania, okay, in one particular plane of division. Here you can see the definite orientation we find. So in this particular orientation, the cell is dividing and producing the two daughter cells. This is longitudinal binary fission in Leishmania. Then we discussed about multiple fission in Plasmodium, and then multiple fission in amoeba. After that, regeneration process in Planaria. Then we discussed about fragmentation in Spirogyra. Then after that, I discussed about budding in yeast and then budding in Hydra. This is all about today's session. If you are new to my channel, subscribe for more sessions. If you liked it, share it with your friends. I'll be back with more sessions. Have a great day. Thank you all.